Okay, so a very swift recap. We know that photosynthesis is a chemical reaction that takes place in plants. It takes place in plants, which allows plants to be autotrophic in terms of their nutrition, i.e. they're self-feeding, they make their own food. If we look at this diagram, we can see here that in order to do this process, we need carbon dioxide, and we need 6 molecules of carbon dioxide to react with 6 molecules of water. Now, if we want that reaction to take place, we need a source of energy, and usually that comes from solar energy, sunlight energy. It can also come from artificial light. What do we do with that artificial light? Well, we absorb it with the chlorophyll, which is in the chloroplasts. We come back to recap that in just a second. And what do? We make 1 molecule of glucose, and we make 6 molecules of oxygen gas. So this is the overall equation. This is photosynthesis, the chemical reaction, and we need to have carbon dioxide, water, we need to have energy, and we need to have chlorophyll inside chloroplasts to power this reaction. We also talked in quite a lot of detail about the structure of the leaf cell and how this leaf is specialized to do photosynthesis. What I want us to focus on, just as a quick recap, is this layer here. This layer is called the palisade parenchyma layer, or sometimes just called the palisade layer. This is where most photosynthesis takes place. This layer is rammed full of chloroplasts, and it's also very close to the sunlight. So as the sunlight comes through here, it's got a very good chance of being absorbed by the chloroplasts within these palisade cells. If light comes through and it doesn't get absorbed, then it's got another chance to be absorbed here down in the spongy parenchyma layer. And the spongy parenchyma layer, also you can see that they have chloroplasts in their cell structure here. What we've got between these cells are air spaces, and those air spaces in this spongy parenchyma layer, okay, can move by diffusion. Gases can move by diffusion out and in through the leaf via the stomata. And the stomata are these pores in the undersurface of the leaf here, and we've got guard cells which are either side of that pore. And remember that the guard cells can change the size of that pore. If you've got any questions about this, please then email me. Make sure that you've got this part of the textbook read and you've got additional notes to support the structure and the function of the leaf and its relationship to photosynthesis. I'm jumping ahead now, but please do look through the PowerPoint presentation of all the things that I've left out. We've done it in previous lessons, and hopefully that's okay. But again, please email me questions, post them in the Google Stream, meet me on Google Meet, etc. But we've got this classic pondweed practical, which is really important for us to appreciate, because what we are doing here is we're measuring the rate of photosynthesis. Remember that rate is the rate at which oxygen gas is produced from that plant, and that rate is a time factor. So we're talking about the amount of oxygen gas that's produced per unit time. Here you've got the pondweed, we've got a beaker of water, and inside that beaker of water we're going to add sodium hydrogen carbonate or sodium bicarbonate. That means that the water here has got plenty of dissolved carbon dioxide. It's obviously got water because it's in water. It's got a light source here, which is artificial, and the pondweed itself has got chloroplasts, which are full of chlorophyll. So it's got all the conditions for the plant to do photosynthesis. And here we can measure the number of bubbles of oxygen gas that are produced per unit time, or we can be more sophisticated and we can do this kind of setup where we actually collect the gas that's produced. This is a much more accurate measurement because we are actually measuring the volume of oxygen produced per unit time, because remember the size of the bubble might vary. And what we can do is we can move the lamp backwards or forwards to measure and see how the rate of reaction depends on the light intensity. We can also put down a filter in this position here, and that filter would change the color of the light. So we can also look at how the color or the wavelength of the light affects the rate of photosynthesis. We might change the temperature, and that will allow us to see how temperature impacts the rate of photosynthesis. And we could also change the mass of sodium bicarbonate that's added here, which would allow us this time to measure how carbon dioxide affects the rate of photosynthesis. So remember that the rate is any kind of change per unit time. We've got this pondweed practical, which is the classic one, which is allowing us to measure the oxygen production per unit time. It's also possible, but it's a little bit tricky in a school laboratory, but we can measure the uptake of carbon dioxide as a measurement of the rate of photosynthesis too. This one is less used, it's an indirect measurement, but this is saying that if the plant becomes larger in biomass, it must have been doing more photosynthesis to generate the nutrients and the energy to grow. So a change in biomass per unit time also gives us an indication of the rate of photosynthesis. And if you haven't already done so, please look at these pages of the booklet and the textbook and make sure that your notes are complete. What we can see here on these graphs are what's called the action spectrum and the absorption spectrum. So if we look first of all at the vertical axis here, you can see that we've got the relative absorption of light. So what we're going to do here is we're going to measure what kinds of light the plants are going to be absorbing. And these A here and B and carotenoids are all different sorts of pigments which are found inside the chloroplast. So the chloroplast principally has got chlorophyll A and chlorophyll B inside what's called the reaction center. And these are pigments found in the chloroplast which absorb particular wavelengths of light. And you can see here that chlorophyll A is absorbing maximally around about here, 450, and also it's absorbing up this end of the spectrum here about 660, and this is nanometers. If you look down here, this is the wavelength, the nanometers of light. So this is measuring the absorption of light for chlorophyll A, and then we've got chlorophyll B, and then we've got this other pigment called the carotenoids. So what I wanted to appreciate here from this top graph is that light is being absorbed here down at the violet blue end of the spectrum very well by this collection of pigments, and then also at this end of the spectrum, 600 to 700 nanometers of light. We're again quite good at absorbing light here, especially through chlorophyll A and chlorophyll B. If we go in the middle, this section here, this is green light. And remember that plants are green because they cannot absorb green light. Instead, the green light is reflected. So that's showing the absorption spectrum of light. And if we look down at this second curve here, what we've got is the action spectrum. So the action spectrum is the rate of oxygen produced per unit time. And you can see that it obviously makes sense because the light that's absorbed here is going to be used for photosynthesis. So photosynthesis works very, very well at the violet blue end of the spectrum. It works very, very well at the orange and red end of the spectrum, but it doesn't work very well here in the middle, the green part of the spectrum, because that light is not absorbed, it's reflected and therefore can't power the photosynthesis reaction. And this graph here is showing that if you increase the light intensity, then the rate of photosynthesis will increase. And we can create a graph like this by doing that experiment with a pondweed practical. So we're going to increase the light intensity. This is the brightness of light. And as we do that, we increase the rate of photosynthesis. But then we get to this plateau stage here. And remember that the reason for this is that light here is no longer the limiting factor. You've got plenty of light, but you haven't got enough of all the other things that are required to keep pace with that quantity of light. And we had that racing car analogy last week, which I loved. It's a really good analogy there. And if we look at the next slide here, we can see we've got a similar shape of the curve for carbon dioxide concentration. So if we increase the amount of carbon dioxide, we increase the rate of reaction until we get to this section here. And at this section here, carbon dioxide is no longer the limiting factor. So you could, in an exam, be given a question with a graph which looks something like this, and you're going to be asked to discuss what's going on at various temperatures or carbon dioxide concentrations. And there's some questions in your textbook which I would suggest that you delve into to have a look at that. So here's the same sort of idea. And if we compare these two graphs here, we can see that the temperature is the same, but because there's more carbon dioxide, this one is increased. So we know that at this stage, this is plateauing here because it has run out of carbon dioxide. Giving it more carbon dioxide jumps it up to here. And similarly, if we compare this one and this one, we can see the difference in the temperature. So just make sure that you can talk through any kind of data which looks like that, and that you can make conclusions based on the data that's shown graphically. So this is the syllabus statement itself, and hopefully we can do this part here. Describe experiments to investigate photosynthesis showing the evolution of oxygen from a water plant. And that's what we've been talking about to date. But then we've also got to look at describing experiments to investigate photosynthesis with the production of starch. So what we need to think about now is how do we measure starch content within a leaf? So this is the protocol here, and when we get back to school, we'll do this together. Again, look at the pages of the textbook here, and what I would like you to do is to do pages 10 to 12 of the booklet within the lesson that we've got today. So the steps are as follows. You take a leaf and you put that leaf into boiling water. And when you put the leaf into boiling water, we can either use a water bath or we can heat it using a Bunsen burner in this kind of way. But what that's going to do is it's going to break down the cuticle on the surface of the leaf. It's also going to break open the epidermal cells, the upper epidermal cells, lower epidermal cells, and then it's going to get through and it's going to open up individual cell membranes. So what we're doing here is we're destroying the leaf tissue and we're opening up the cell membranes. The next thing is to take that leaf and place it into a boiling tube of ethanol, and the ethanol needs to be warmed up. Now, we've got to be careful because ethanol is flammable. So at this stage, it would probably be better to make sure that the flame is off and the water was hot, or to use a water bath. But the purpose of this stage here is that the ethanol is a really, really good solvent of chlorophyll. So the chlorophyll within the leaf, which is now the cells are open so we can get at the chlorophyll will be dissolved, the ethanol will become green, and the leaf becomes a very sort of grayish white color. We take that leaf then and we wash it. We wash off all the excess ethanol, and also by this ethanol treatment, the leaf becomes quite brittle. But by placing it in here, we soften the leaf up and we wash off the excess alcohol and we place it on a white tile. Then we're going to add this chemical here, which is starch. Sorry, not starch, it's iodine. And iodine is the test for starch, as you know from previously in the course. So if we got iodine on the leaf and the leaf remains brown, that means there's no starch present. If the iodine goes black, that means that it has got starch present. And if it has got starch present, then that means that it must have been doing photosynthesis. Because remember, when the plant does photosynthesis, it initially makes glucose, and then the glucose molecules are individually put together to make an insoluble storage molecule called starch. So these are the steps that are necessary for testing the leaf for starch. And then what we can do is we can put plants in all sorts of different conditions. We can change the carbon dioxide levels that the plant has got. We can change the intensity of the light. We can change the wavelength of the light, and we can run through these particular tests. One thing that's important before we do this is that we normally need to de-starch the leaf. So before we set up an experiment, normally we put the plant in the cupboard, and in the darkness, it has no access to light, which means it's going to break down all of the starch which is in the plant. So when we start the experiment, the plant doesn't have any starch in it to begin with, and then we would set up a variety of plants, possibly at different light intensities or different wavelengths of light, or having access to or not having access to carbon dioxide. And if you look at the booklet, there are various experiments which are set out there, and we will do these when we come back together when school reopens. This next slide here is just taking you through the steps as a quick recap. This slide here, this YouTube clip is also in our Google Classroom, and it's a really, really good clip on limiting factors, which I would like you to go back into and to reinforce before you look at the next video. Thank you.